Hello and Assalamualaikum everyone. Today we are going to present about what did we get based on our case study. So I will start first. My name is Nurain Hazratul Raihana. My matrix number 230955. Today I will present case report 1 which investigate the pathogen responsible for a fatal lung infection using protein blast or blast P. In this case, a patient from Hospital Tuaran Sabah died after developing severe respiratory symptoms following exposure to a suspicious powder contained in a letter. Three unknown protein sequences were isolated from the patient lung sample and analyzed using BLAST-P to identify the protein and determine the most likely pathogen. Our BLAST-P analysis showed that protein A matched a set domain containing protein or protein lysine and methyltransferase from Clostridius difficile with 100% of identity. This protein is involved in regulating protein activity and gene expression. For protein B, it matched fucus binding lectin-2 from Burkholderia caenocepacia, also with 100% of identity. This protein helps bacteria to attach to the host cell, which promote biofilm formation and contribute to the infection. Finally, for protein C, it matched the need domain containing hemophil IXDX1 from Bacillus anthracis with 100% of identity. This protein plays an important role in acquiring iron from the host, which is essential for bacterial survival and virulence. Although the three species matches different bacterial species, the patient clinically history strongly suggests that Bacillus anthracis is the most likely causative pathogen. This is because the patient was exposed to a suspicious powder-filled letter and developed severe lung symptoms, which are characteristic of inhalational anthrax. In addition, the identification of ISDX1, a non-virulence factor of Bacillus anthracis, further support this conclusion. In conclusion, this case demonstrates that BLAST-P is a valuable bioinformatic tool for identifying unknown protein. However, the final diagnosis should not rely on sequence similarity. Combining protein analysis with clinical evidence provide a more accurate identification of disease-causing pathogen. That's all for my part. I will pass the presentation to Wawa.
Good evening, everyone. Today I will present our case study 2 about PHI blast analysis. The aim of this case study is to identify an unknown protein and find the most likely bacterial pathogen using PHI blast. First, let me explain the case. The victim is Mr. Rodney. He had symptoms such as fever, vomiting, and shortness of breath. A suspicious white powder was found in an envelope together with a blood-stained note. The note contained the protein excision number OON704171.1 and the motif. To identify the protein, we first got the protein sequence from the NCBI protein database. Then we run PHI blast using the motif. After that, we check the alignment, verify the motif, and identify the protein. Our result show 100% sequence identity and 100% query coverage, which means the math is very accurate. The motif was also successfully verified. Based on the result, The unknown protein was identified as a set domain containing protein. This protein had bacteria by adding methyl groups to other proteins, which is important for bacterial function. The analysis also showed that the bacteria most likely belongs to Bacillus cereus group. This group includes Bacillus cereus, Bacillus anthracis, Bacillus thuringiensis, and several related species. However, PHI blasts cannot tell us the exact species because they have very similar protein sequences. In conclusion, PHI blasts successfully identified the unknown protein and confirmed that the pathogen most likely belongs to Bacillus cereus group. So that's all for me. Thank you.
Okay, next, I will present our third case report on position-specific iterated blast, blast, which was used to identify the unknown protein X. In this case, Mr. Rodney became seriously ill after being exposed to a suspicious white powder. During the post-mortem examination, an unknown protein X was isolated from the tissue sample. Therefore, blast was used to identify the protein and its possible origin. The amino acid sequence of protein X was searched against the NCBI non-redundant NR database using 3 blast iterations. The analysis showed highly significant matches with set domain containing proteins, with the closest matches being Neobacillus vireti, Neobacillus cucumis, Neobacillus drantensis, and related Bacillus species. The sequence alignment also showed high similarity and a conserved KKGELIHAAP motif, confirming that protein X belongs to the set domain protein family. Based on these findings, protein X was identified as a set domain containing protein, specifically a protein lysine and methyltransferase with the highest similarity to Neobacillus vireti. However, it was identified as a protein domain rather than a bacterial toxin. So, the evidence was not sufficient to identify a definite pathogen or conclude that Mr. Rodney was murdered. That's all from me, thank you. And I will now pass the presentation to the next presenter.
Assalamualaikum everyone, my name is Hazika Izati binti Saki Ilais Roslan and my nometic is 228194. Today I will present about case 54. So the objective of this case is the identification of unknown isolated DNA to T plus N and phylogenic analysis. So regarding the case overview, the case present where the patient have acute lung poisoning and where the patient have present with 2 days of history of vomiting, nausea and short breath, and also due to the dubious letter that opened prior to the case with suspect of patient inhalation poisoning. And prior to death, patient has uttered a 6 of 6. Then, autopsy, it was noted that the patient have extraordinary inflammation of lung and accumulation of yellow mucosal membrane on patient. And from this mucosal membrane, protein S was obtained and isolated and sequenced. And regarding of the research flow, the porting S was blast to the T blast N, where the 6 suspected pathogen was selected and go to multiple sequence alignment, and then we will generate and analyze the phylogenetic tree. So from the phylogenetic tree analysis, it was found that the Bacillus anthracis is with the with the Bacillus cereus has a 99% of bootstrap, which indicates high statistical confidence, including that uh, including that it also was found that the branch both of these share a common ancestor, which means that have a short form of evolutionary relationship. Then From what we can conclude from the phylogeny tree is that the phylogenetic analysis suggests that Bacillus anthracis is a casualty a pathogen with a 99% boosted support, revealing an extremely high confidence in evolutionary relationship despite the Bacillus series share the same bootstrap value as a sister species. This is due to the absence of the virulence plasmid that suggests is less likely to be the causal pathogen, and similarly the clinical symptoms such as short breath, nausea, and vomiting directly match with the inhalation anthrax. That's all for me. I will pass on for the next presenter.
Okay, so thank you. I'm going to present about case file number 5. So to begin my case, I would like to present the overview of the case. So the case revolves around Mr. Rodney, who has found a strange letter containing white powder 2 days ago. Unfortunately, this has led to the death of Mr. Rodney after 2 days. During the 2 day period, he has developed symptoms like shortness of breath, continuous vomiting, high fever, and so on. And according to Mr. Rodney's assumption, this is due to infection with K pneumonia. However, he couldn't definitively identify the pathogen. And also, he has left two notes as clues, saying an accession number and also a pattern sequence. So again, what we know from this incident is that white powder was found and the patient experienced symptoms such as shortness of breath, high fever, and vomiting, and could have been infection of K pneumonia according to the patient. And the patient has left a sequence pattern and an accession number as a clue. So we have to find out about the sequence to find out about the sequence and determine the pathogen by carrying out fibroblast analysis. So we have to determine the inputs and using protein blast by using the accession number as the query, and also select fibroblasts to determine the pathogen. Right. So next, let's move on to the results that we have obtained from protein sequence analysis. So from the protein sequence analysis, we have received top 4 sequences that showed very high level of similarity in sequence, where the expect value is near zero value, meaning that the matches are strong, accurate, and has not happened randomly. And the identical amino acids are also found in the same position among all the 4 sequences that have obtained in the results. So now let's look at the 4 sequence to identify them. So sequence 1 is, so all 4 the sequence is SET domain containing protein. First sequence is Bacillus cereus. Second is Bacillus parenthesis. Third sequence is Bacillus pacificus. And sequence 4 is Bacillus anthracis. Right. However, from the 4 sequences, we have successfully identified Bacillus anthracis as the cause for the death of Mr. Rodney. This is because Bacillus anthracis is a bacterium that forms fine white powder, which is often used as a biological weapon to attack people. So, and the symptoms of this pathogen infection also is similar to what Mr. Rodney experienced. That is shortness of breath, vomiting, and fever. And the death is very quick, which can happen within 48 hours. So, as a conclusion, we have concluded that this is the pathogen that has caused the disease via fibroblast, where 100% sequence identity was obtained. So from the matches, we know that top 4 matches were found, and through cross-reference to the clinical and forensic data, we can finally conclude that Bacillus anthracis was the pathogen. That's all for my presentation. Thank you.